Hello, my name is Linda Ebbert and I am a research associate at the Institute for Medical Informatics in Münster, Germany. Today I would like to present tRuffle, an iRNA app for exploring unique differentially expressed genes in single-cell RNA sequencing clusters. I would like to start with some words about single-cell RNA sequencing data. This data is generated by single-cell RNA sequencing. This method generates high-dimensional data, and that's why a dimensional reduction is an essential step for the analysis of this kind of data. You can see the results of the clustering step on the right side by looking at the UMAP or t-SNE plot. Clusters often represent cell types that can then be further analyzed and compared. For the expression profiling of one cluster, you often compare it to all other clusters at one step, resulting in differentially expressed genes for each cluster in one table. This data is the correct input for tRuffle. 5 columns are required and need to be specified right after uploading the table in Excel format. These columns should comprise the gene, its p-value, its adjusted p-value, the log2 fold change, and the cluster it belongs to. Here you can see a main view of tRuffle. After uploading the data, you are now ready to get some filtering done. You can set thresholds for the p-value, the adjusted p-value, and the log2 fold change to filter your data. By looking at the table and adjusting the sliders on the left side for each value, you will see it changing dynamically. Have you already noticed the last slider? Here you can adjust how unique your differentially expressed genes need to be. The uniqueness score considers the total number of clusters and the number of clusters containing a differentially expressed gene. A score of 0 would then represent differentially expressed genes found in all given clusters, while genes that are differentially expressed in a single cluster will be scored as 1. That means by setting the threshold to 1, you would just get cluster-specific differentially expressed genes. So, now you filtered your data, but maybe you still wish to get a better overview. This upset plot is generated by the R package upsetR and shows the intersection sizes between your chosen clusters. Here you should have in mind that all thresholds you set before are still active. Also the threshold for the uniqueness score, so be careful. By looking at the horizontal bars, you can see the set size of differentially expressed genes for each cluster. The black dots mark the clusters that form an intersection, and the vertical bar above them represents the intersection size. Taking a closer look at those vertical bars, you might notice that cluster 5 and cluster 1 seem to share almost 60 differentially expressed genes. Maybe the cells of these clusters have a common progenitor, or they might have similar roles for the tissue function, or something else. So, now that we chose an interesting intersection, we can select cluster 1 and 5 and take a closer look at the genes that we found. By clicking on the Intersection DEGs tab, you finally come to your meaningful and rare genes, your personal tRuffles. Here you can check out the significance values for the genes that are only present in cluster 1 and cluster 5. To go on with further analysis, for example gene ontology, you can easily download your results as a common Excel file. So, now let me quickly sum up the advantages of using tRuffle. By setting this threshold for the uniqueness, you can adjust your filtering for varying levels of technical noise and biological variance. In addition to that, you can explore cluster-specific genes or those that are specific for a small cluster group. tRuffle is very easy to use and has an intuitive graphical user interface. It is a fast and adaptable tool to generate valid biological hypotheses from single-cell RNA sequencing. And last but not least, it is very easy to download your results in Excel format. Thank you so much for listening. If you have any questions, then please feel free to speak to me at the ISMB or just write an email.